We're back with a shorter topic today. Today we're going to look at taxonomy. That's how we classify living things, specifically with an eye towards phylogeny. Phylogeny refers to the evolutionary history of organisms and their evolutionary relationships with other organisms. Taxonomy is simply the art or science, I suppose, the process of taking categories and arranging them in a hierarchy. Now, what I mean by that is you take a small category and you make it part of a bigger category, and you make that category part of a bigger category, etc. It's kind of like those Russian stacking dolls that you might be familiar with. We've got a little doll that fits inside a bigger doll that fits inside a bigger doll, etc. That's a hierarchy. Now, this is a system that doesn't just apply to biology. It's a system that we use all the time in our everyday lives, even if you might not think about it. Imagine you're a big fan of the author Stephen King. You go to the library and you might find a whole shelf of Stephen King novels. Man, that guy writes a lot of books. And that shelf might be part of an entire bookshelf that's made up of novels from the horror genre. And of course, this bookshelf is probably going to be located in the fiction part of your library. So categories within categories. In modern day biology, when we make categories, we always try to make sure that the organisms in those categories are related to each other. This is known as systematics, and it's a type of taxonomy. This is where we're trying to make sense of biological diversity by making categories, and we're doing that in an evolutionary context. Ideally, taxonomy done this way through systematics should be based on a phylogenetic tree. Phylogeny refers to the evolutionary history of an organism and its relationship to other organisms. And a phylogenetic tree is a branching structure that shows the relationships of the organisms on the tree. You're seeing a rather famous and rather old phylogenetic tree to the right here. Now, what do we see on this tree? Well, we see relationships between different organisms, and also we see that we have older, simpler organisms at the bottom. Now, this tree has a few problems by today's standards. First of all, it's really biased towards humans. We have humans, as the pinnacle at the top. It kind of harkens back to that great chain of being that we talked about in the very first lecture. We try to avoid that. Also, we typically today will not put different organisms on the trunk of the tree. They only go at the ends of the branches. And I'll explain why that is in a little bit. Finally, there are some actual problems with these relationships. Some of them have been proven to not be quite right. But I thought I'd start out with just a very old and very famous phylogenetic tree, and we'll see if we can tidy that up a bit. Screws and bolts. There's a lot of them. They come in all different lengths and sizes. Bolts have a flat end at the end and they go into a nut, and screws have a pointy end so they can drive into something. They have different heads on them for different types of screwdrivers and ratchets. They're made of different types of metal, and they're made to go into different things. So, if you've ever been out shopping for screws, you know that if they were not sorted, if they were just thrown together, you would never find what you're looking for. This is an example of taxonomy, what we can do to make sense of these screws and put them into categories and into little bins. This is not a case of systematics, though. I just want to point that out because, of course, screws are not living. They're not evolving. Here's an example of how we might sort some screws. We've got 4 rows here, and we've got different types of screws. Some that are meant for drywall, some that are meant for wood, some that are meant for metal. Tex is a certain brand name that will self-tap or drive itself into a lot of different things. And then we could arrange them along that row into bins and sort them by size, etc. There's a lot of taxonomy going on at your local hardware store. Here we've got a whole lot of screws and bolts, and they've been sorted based on what you're going to use them for: metal, wood, whatever, concrete, etc. They might be sorted by the metal they're made of. Are they made of brass? Are they made of zinc, etc.? They could be sorted by what type of screwdriver you're going to use. They could be sorted by length and by width, etc. So this helps us make sense of a very big mess. And again, that's the whole point of taxonomy, and that's why I'm using this screw example. It gets even more messy though. Just keep that in mind with living things, because there we're talking about millions of species. My last slide of screws, I promise, and then we'll get into some actual living stuff. Sometimes when you're trying to build these categories, it's useful to use a system like this. A system of questions, one after the other, and each question has a yes or no answer. There's only two possibilities. And when you have something like this that branches in two, we call that a dichotomy. All right, so let's work through this. The first question on the left there: is the top of the shaft threaded? Then we have a choice, yes or no. That might be hard to see, the yes or no, but it is there. If it's yes, we go up. If it's no, we go down. Let's say it's no, let's go down. The next question is there an X-shaped slot in the head? And again, we have two choices. We have yes, and we have no. So let's say that the answer is yes. The next question is the head domed? So is this a screw with a dome-shaped head? And then we have our question. We have two possible answers. Yes, we go up, and it's a Phillips roundhead wood screw. Or no, it's a Phillips flathead wood screw. Now you may have come across something like this before. If you are interested in birds and you have a bird identification book at home, let's say, quite often in the back of that book there will be a series of questions like this to help you identify the bird that you're trying to find. Well, it also works for building phylogenetic trees, as we'll see. All right, let's leave the screws behind and move on to some living stuff. We've seen this guy before. This is Carl Linnaeus. And back in the 1700s, he gave us an excellent system of taxonomy for living things. He also created a naming system that we still use today to name species. Now, he was not an evolutionary biologist. He was a natural theologian. He was classifying God's creations. And he picked some really good characteristics to make his groups. In fact, many of these characteristics are still used today. They still hold up coming at this problem from a systematics approach. For example, he looked at whales and he recognized that they were not fish. Most people in his day would have called them fish. But he recognized that fish had tails that went side to side. He recognized that whales didn't. Their tails went up and down. Whales, of course, have blowholes. They breathe air. Fish don't. And whales produce milk. That's a characteristic of all mammals. Also, he may have noticed that some whales have tiny hairs on some parts of their body. Back to that naming system. It's called the binomial naming system. Bi means two, nomial means name. So it literally means two-name naming system. And as an example, I'm sure you've seen this before, this is our proper name, Homo sapiens. So he decided to name all species, and he named a lot using Latin. The reason for this is Latin is a dead language. No one's going to get upset by that. If we put it in French or English or something like that, some people might get bent out of shape by that. Also, he realized that we needed to have one agreed upon name for every species so that biologists from around the world could talk about them. There's different common names, of course, for different species everywhere in the world, but if we can decide that this name in Latin is going to be the name, then there shouldn't be any arguments. So with this binomial system, the first part, so Homo in this case, is the genus. The second part, sapiens, is called the specific epithet. There may be several species or a handful of species in a genus, but the second part is specific to just one species. So again, the proper name for humans using this system is Homo sapiens, and translated that means wise man. Linnaeus named 12,000 species, which is pretty remarkable. And we still use almost all of those names. He named some large groups as well: aves, the birds; amphibia, amphibians; insecta, the insects. We still use those groups. Some of his other ones we don't use in a formal sense anymore. Paradoxa is kind of interesting. They're a paradox. These are things he couldn't fit in anywhere else. It's interesting that today we have a similar group still. We have something called the Protista, which we will talk about later, which are also things that we can't fit neatly into other categories. It is remarkable, though, how well his basic system has stood up. So what categories, what groups are used today? Let's start at the top with the biggest, most all-encompassing group, the domain. There are 3 domains: Eukarya, Archaea, and Bacteria. These domains are separated from each other based on cellular structure and biochemistry. So these 3 groups are fundamentally different from each other. And I'm just noticing for the first time that Pearson Education seems to think that there are 4 domains. There are not. There are 3. If we take a look at that critter down the bottom there, that panther, Panther pardus, that's the particular species, let's work out his whole classification scheme. So we'll start at the top. That panther is made up of eukaryotic cells. So it's in the domain Eukarya. Now within the domain Eukarya, there are 4 kingdoms. Hey, Pearson messed up again. Anyway, he is in the kingdom Animalia. Within that kingdom, we have several phyla. So a phylum is a smaller group within a kingdom. He, and also us, is in the phylum Chordata. He's in the class Mammalia and the order Carnivora. So we're getting smaller and smaller and smaller with our categories here. The mammals, as we just talked about, they're defined by having hair and producing milk and breathing air and live birth and a few other things. Carnivora are defined by their teeth and a few other things. Within the order Carnivora, we have a bunch of families, and this individual is in the family Felidae along with the other cats. In the genus Panthera, there's a few other panthers out there, and again, the specific species is Panthera pardus. So this is how we do our classification. And once again, this is based on systematics. So for instance, Felidae, that includes all the cats. They have a shared ancestry. As we go down, the shared ancestor becomes more recent. Now let's look at a tree here, a phylogenetic tree. And here we're just looking for members of Carnivora. So we have our families here. We have Felidae, the cats. We have the Mustelidae, which are things like weasels and otters and so on. And we have the Canidae, which are the wolves and the dogs. We have our panther there, so you can see this time we're going up, order, family, genus, species. And you can see that in this tree here, these groupings that we've made are based on relatedness. So we have the skunk and the otter, and they're grouped together in this Mustelidae because they have a common ancestor at that branching point. When you're building a phylogenetic tree, you can use big groups with lots of organisms in them, like you can see on the right here, or you could use individual species, like you see on the left. So we could have branches or twigs. Well, no, actually, they're all called branches. I'm just trying to be funny. Let's start with the diagram on the left. We've got 3 species of Drosophila. Because they're all in the same genus, the genus Drosophila, we know they're very closely related, and they would have separated from each other. They would have speciated not very long ago. Now note that the name there, we can take Drosophila and shorten it to just D period. So that's something you'll see quite often. It's just, makes the words a bit shorter and more manageable. One thing I didn't mention before is that when you're writing scientific names, the genus is always capitalized, and the specific epithet, the second part, is never capitalized. Now what they should have done here is written this in italics as well. They didn't do that, but you should do that. You should type it in italics. That's just something you do if you have Latin text. Anyway, let's take a look at this first phylogenetic tree, this very simple one. There are 2 dichotomies. Remember, that's where branches split off. Another name for a dichotomy is a node. So we have node number 1 and node number 2, and those are speciation events. So at node number 1, we had a population splitting into 2, and then one of those branches became Drosophila obscura. The other branch became the ancestor of Drosophila dentissima and Drosophila melanogaster. So at node 2, that's where those 2 species split. So you can see we can get a lot of information from this figure. We can start at the bottom, we can work our way up, and we're going forward in time. Or we can work the other way and we're going backwards in time. One thing to note is you would never put your species anywhere but at the end of the branches. This will become more important in just a moment. We wouldn't put them on the nodes because that would suggest that we know that a certain species is the ancestor of another species. And we very rarely would know that. We can't say for sure what species gave rise to what species. So we only put them at the end, and we're looking at relationships here. So don't put the species names at nodes or on the branches, put them at the ends of the branches. Now, having a quick look at that other phylogenetic tree, this doesn't follow that same format. The one on the left is something called a cladogram, and we'll talk about that in just a second. That's the best way to do a phylogenetic tree. The one on the right's a little sloppier, but the one on the right, you can see that they've got very large groups of organisms. Animals, there's thousands of animals. There's lots and lots of plants, etc. So I just wanted to point out that a phylogenetic tree can show how large groups are related to each other or individual species. Alrighty, so how do most modern day biologists go about generating phylogenetic trees? The majority use a system known as cladistics. In cladistics, what you do is you look for shared traits or characters or characteristics. The characteristics that you're using are characteristics that are shared within that group because of a common ancestor. If we're doing this correctly, we refer to the groups that we make as clades, and clades should be monophyletic. And what the heck does that mean? Mono means one, phyletic, that refers to phylogeny. So again, phylogeny is the study of ancestry. It's the study of evolutionary relationships. We want to make sure that every single group that we make, every clade that we make, can be traced back to the most recent common ancestor that had all of those traits. So that ancestor evolved those traits, and then it branched out and gave rise to the other species or groups that we see in our clade. When we do this, the phylogenetic tree that we create is known as a cladogram. What you're seeing here is a cladogram for primates. We, of course, belong to this group. On the left there, we have the common ancestor of all the primates. So I've shown that as a blue dot. This is a dichotomy, also known as a node. But we can take lemurs, New World monkeys, etc., through to humans. We can trace them all back to that common ancestor. This is the last common ancestor, the most recent common ancestor, that had all of the traits that we're using to define this group, the group primates. And quite often we'll label the group at that node, at that dichotomy. As we move towards the right, we add in more characteristics, more traits that we can use to define subgroups. So we have the primates, but there's apes. Apes are a subgroup of primates, and the great apes are a subgroup of the apes, which again is a subgroup of the primates. So this node here, this dichotomy, represents the common ancestor of the apes. Gibbons, orangutans, gorillas, chimpanzees, humans, they're all apes. They can all be traced back to this common ancestor. And if we go a little bit further to the left, we have the great apes. Orangutans, gorillas, chimpanzees, humans, these are referred to as great apes. It's based on teeth and a bunch of other things, but we have characteristics that define that group. We're excluding the gibbons now, and actually they're usually referred to as lesser apes. But this is the way a cladogram works. We are adding new characteristics as we move towards the right. We're defining new subgroups, but each group has to be traced back to a common ancestor. So how do we do this? Well, the system was invented by this guy, Willy Henning, and after he came up with this, it kind of took off, and like I said, it's the most common means of recognizing these relationships and building groups. So a few terms here. What we're looking for are referred to as characters. Characteristics. Same thing, but in cladistics they're usually referred to as characters. And what we want to do is pick one character that we can use at a dichotomy, at a node, that we can form a question with. And that question should have a yes or no answer. So for instance, for mammals, is milk produced? Yes or no? Is there fur? Yes or no? So those are the kind of questions, those are the kind of characters that we're looking for. So the character can either be there or not be there. So it's either absent or present. We're trying to identify primitive characters. These are known as plesiomorphic characters, but we're not going to bother with that term. It is a better term in some ways because primitive has kind of a connotation that goes along with it that, you know, if you've got these primitive characters, well, you're somehow worse, but we'll just stick to primitive character. This is a character that appeared much earlier in the development that led up to this group that we're trying to study. And we have derived characters, and these are what we're most interested in. So these are characters that popped up just in the group that we're trying to make. The ones that we want, like I just said, are the ones that are shared within a group. So these are called shared derived characters. So again, the ability to make milk would be a shared derived character for mammals. However, the presence of a spinal cord, that's not something that's only found in mammals. So that would be a primitive character. And that's one that's not going to be too helpful because there's lots of other things that have a backbone. So we're looking for shared derived characters that we think are only going to be present in our group. That's how we're going to build our group. So let's have a look at how we might use this. We'll take a look at a really simple example here. So we've got some mammals here, and then we have a turtle, which of course is not a mammal. We refer to the turtle as the outgroup. It's almost like a negative control in a way. We're trying to define different groups within the class Mammalia. And if there's something that's also present in the turtle, which is a very distant relative, then we know it's a character we can ignore. Like, for instance, a spine. Whether or not an animal has a spine is not going to help us sort out the relationships within the mammals, right? Because, fish have spines and reptiles have spines and birds have spines. It's not that useful for what we want to do, which is just simply figure out all the groups that are within the class Mammalia. So we need to look at derived characters. So the cladogram will allow us to build this hierarchy of groups, like we've talked about quite a bit, and it's going to be based on shared ancestry. We're using just derived characters. And again, the outgroup is something that we can compare to. So going through this from the bottom, we can see that between our branch that leads to turtles and the horse, we have the appearance of hair. And that little red line is going to be used to show the origin of this particular trait, which is hair. And then we see between where the horse comes off and the wolf, we have the development of carnivorous meat-eating teeth. And then between the next two nodes, we have retractable claws. And then the next one's not great. The ability to purr, we're using that to identify domestic cats, although, like I said, it's not great. There are some other cats that could probably purr. But anyway, looking at what this means, if we go way back to where the hair appeared, that's something that appeared in the ancestral mammal. It never appeared in the branch that gave rise to the turtle. So that is a defining feature for all mammals. We go up a little further, we see that we have carnivorous meat-eating teeth. We didn't see that in the horse. They branched off before that developed in this lineage, but that is a character of all of the carnivores. And then retractable claws, that's something that just defines the cats. We don't see that in the wolf. So this is a nice simple cladogram, and this is the way you would draw one. And here are the general parts of a cladogram. At the base, we have the root. Again, think of this as a tree. We have the nodes, and that's where we have two branches splitting off. And remember, it should be a dichotomy. It should be two branches. We don't want more than that. At the top, that's where we put our actual groups or taxa, or if we want to be really proper, they would be clades. We put those at the top. We don't put them anywhere else. And this is the case even if we're dealing with things that are extinct. So if you have a fossil species, you have to avoid the temptation to put it anywhere else. If you put it at a node, then you are making the bold claim that that fossil species gave rise to the species above it. And you can't say that. We don't know that. Just to drive that point home, here's a cladogram of some dinosaurs. Dinosaurs are, of course, extinct. Well, except for that one dinosaur up in the right top-hand corner. Yes, birds are considered dinosaurs. We'll come back to that. But you have to avoid the temptation to put fossil species anywhere else other than the ends of the branches. You can't put them at the nodes. You can't put them in the middle of the branches. This is not a diagram that shows us this species evolved into this species, evolved into this species. It doesn't work that way. This is a diagram that shows us relationships. Relationships between species that are extinct, relationships between species that are alive, and relationships between species that are extinct and species that are alive. And just to draw your attention to the fact that there is more than one way to draw a cladogram. I think the one on the bottom is the easiest one to draw, and I am going to have you draw some of these. I'm going to have a little assignment. You'll see this on tests as well. But notice the one up above that, it's really exactly the same. It's just laid out a little differently, but it shows the same dichotomies. Let's have a look at how we would go about constructing a cladogram from scratch. We have some very different groups of animals here, and all of them but one are vertebrates. That means that they have either cartilage or bone surrounding their brain and helping to support their spine and protect their spinal cord. The outgroup here is something called a lancelet. It is not a vertebrate. It is related to these guys, but it doesn't have those structures. It doesn't have anything resembling a skull or anything resembling a vertebral column. So we're going to use that as our outgroup, and we're going to compare that to the other organisms here. If we find a characteristic that's shared by everything, including the lancelet, we're going to ignore that because what we're interested in doing is sorting out the relationships just among the vertebrates, the lamprey, tuna, salamander, turtle, leopard. And anything that's also found in the lancelet isn't going to help us. That's a primitive characteristic, not a shared derived characteristic, which is what we're looking for. Okay, so we have on the left here something called a character table, and that is where you would start. So what you do is you look for the character that's found in most organisms. So imagine we didn't already have this cladogram here. We're just looking at organisms and we're taking some notes. And we find that all but one have the vertebral column. We know that this is the very first character to appear. We have that in 5 of our 6 organisms. Now next we have jaws. We see that in 4 of the 6, so we know that's the next character to appear. The walking legs, we have that in 3 of our 6, so that's the next character to appear. The amniotic egg, so that is found in 2, and that would be the next character to appear. And then hair, which is a defining feature of mammals. Now, one thing you might be scratching your head about is the amniotic egg. Turtles lay eggs, sure. But do leopards lay eggs? They don't. But something we'll talk about much later is the fact that inside a mammal, we have all of the same tissues that we would have inside an amniotic egg. An amniotic egg is something like a reptile egg, a bird egg. These are eggs that have a hard shell, and if you crack open the egg with a developing embryo in it, the embryo has an amniotic sac around it. There's a yolk sac. There are blood vessels that lead to a little cord that go back to the yolk sac. All of these tissues and structures are internalized in mammals, as we'll talk about much later. So we do have those same structures in mammals, as weird as that might sound. But you can see you start with your character table, and we've used zeros for the absence of the character, so we don't have the character, and ones for the presence of the character. These are things that computers like. They like ones and zeros. They work on ones and zeros. And once you have a character table like this, you can feed that information into a computer, and it will spit out a cladogram for you. And it will try to make the simplest cladogram it can. There's a concept in science called Occam's razor, where we start with the simplest explanation. And we test that first and see if it holds water. That's what the computer will do for you. Now, if you're doing this kind of analysis, I mean, this is extremely simplified here. You wouldn't use just one character, you would use a bunch of characters. So you would feed in tons of data into a computer. It would look at all that data. It would use ones and zeros, like not present or present, and then it would spit out a bunch of cladograms for you, and it would try to find the simplest one that explained what you were seeing. A lot of cladograms nowadays are made using DNA sequences or sometimes protein sequences, and it works the same way. So you can find a DNA sequence of a particular gene for the organisms that you're interested in. You can get more sequences for the same gene from databases online, so scientists share all this stuff. And then you can line up those DNA sequences, so align them and look for differences that have developed over time after these organisms separated from each other. So what you're seeing at the top here are two identical sequences of DNA. So perhaps this is all from one population of one species. The populations separated, the species separated into two species, and now they're separate and there's no gene flow. We're going to get some mutations. We might get deletions, so we might get nucleotides just being cut out and disappearing. We might get insertions, so we might get DNA inserted. So imagine that that happens, and now our DNA sequences don't line up quite right anymore. But you can run that through a computer and it will look for the matching pieces and then look for the differences. So down the bottom there, number 4, we're comparing these two DNA sequences long after these species have diverged from each other and changed. And we can see there's one gap there where we have GTA, that's a sequence of nucleotides, in number 2, but we don't have that in number 1. So that's a difference. And we can just treat that exactly the way we would the presence of hair or the absence of hair. We could just code this as the presence of the GTA or the absence of the GTA. Likewise, we can see that we lost that G. So we have on the bottom here, we have a G that doesn't line up with the same spot on the other species. And again, we could code that in our table as, is there a guanine present or is there not a guanine present? It's the same process. Let's take a look at a more realistic example. We've got 9 species that we want to compare. We want to figure out how they're related to each other. So we take a sequence of DNA from the same gene for each of the 9 species. We have a 10th species, which is going to be our outlier, our outgroup. A computer can take those sequences and line them up, find a best fit, a best match between those species. So line up the nucleotides one by one and then look for gaps. The gaps are where nucleotides were lost through mutation, through deletions. Now looking at what we have down the bottom there, the first thing we want to hunt for is the change that's shared by most of the species. And it would look like that is that blue gap. So that's where we're going to start. So here we can see that our first node, our first dichotomy, is between one lineage that has lost that nucleotide, that T, and another lineage that has not. Now the computer would have run a whole bunch of different scenarios and it would look for the simplest explanation. That's where we always start. So it would look for the cladogram with the fewest steps. And the computer has decided that the simplest explanation is that those T's were lost in everything but number 10, and then they were regained later on in 8 and 9. Computers tend to try to avoid that because that's not something that happens too commonly, but it was the simplest explanation. Now we can see after the next dichotomy, we have that big gap where a lot of nucleotides have been lost, the one that's colored green, that's popping up. And of course, that character, the character of missing that sequence, has been passed down to 8, 9, 7, 5, and 6. So I won't go through all the bits and pieces of this with you, but feel free to pause the video and have a look at this and see if it makes sense. Again, the computers are great for this because they can test many, many different scenarios. And what a computer would do if we were doing this in a real analysis is next we would give it a different DNA sequence and then a different one. And we might test several different genes and then have the computer crunch all that, put it all together, and try to come up with the simplest explanation for changes in all those different genes for all these different species. Again, though, the main point is that we can use DNA evidence sequences in exactly the same way that we would use other traits like hair or the production of milk. Let's go back and look at some physical traits again. And this time we'll talk about what we shouldn't do if we're making a cladogram or if we're making any kind of phylogenetic tree. Let's start at the bottom here. We have a shark, and then next to that we have an orca, a killer whale. And superficially, they look pretty similar. They have a similar body shape. And the reason they have a similar streamlined body shape is because they need to travel through the water, and that shape works really well. They also have pectoral fins, which goes way back. That's something that evolved much, much earlier. It's a very primitive trait. It's not that helpful for us. But there are big differences. We have a tail that goes side to side in the shark. It's a fish. We have a tail that goes up and down in the orca. It's a mammal. The orca breathes air. The orca makes milk. We've talked about all those quite a few times. So if we were just to use the body shape as one of our characters for a cladistic analysis, we would get bad results. We can't ignore all of that other stuff. The shape is simply the result of analogy. If things are analogous, that means that they look the same because they perform a similar function. They have to have a similar shape. It's like a bird wing and the wing of an airplane. They have to have a similar shape to provide lift. That's how flight works. What we want to find are homologies. We want to find shared derived characters that are common in a group because that group had a common ancestor. So looking at the top now, if we compare the killer whale to a dolphin, they share all those mammal characteristics. And they share those mammal characteristics because those are homologous. Homologous traits are traits that are shared between two organisms because the organisms have a common ancestor, and the ancestor had those traits. So the ancestor of the killer whale, the ancestor of the dolphin, they were both mammals, so they had those mammalian traits. So we don't want to go by the shape in this case. We want to go by those traits that are shared because of a common ancestor. And I mentioned Linnaeus before. Even though he wasn't looking at things in terms of evolution, he wasn't thinking about common ancestors. That's not the way he was approaching this. But he still realized which traits were important. When we're doing phylogenetics, we have to be really careful about something called convergence. This is where we have two species that are not closely related. They do not share a recent common ancestor, but they might look very similar. And they look similar because they have a similar lifestyle. They have to do similar things. They have to have similar organs or similar shapes, again, because of analogy. They have evolved analogous structures that do similar things and look similar, but they've evolved those completely separate from each other. A really nice example here is an extinct reptile known as an ichthyosaur. It looks a lot like a dolphin, but it's a reptile. It's not a mammal. And you can see there are some differences. So once again, the tail goes side to side, which is interesting. But also, if you look at the fossil at the bottom on the right, the spine goes into just one fluke, one part of the tail. That's really weird. That's very different from dolphins. It's quite different from fish as well. And if we look at their flippers, their paddles, we can see that they have rear limbs still. Modern whales do not. They have the same bones, but they're arranged quite differently. They have the humerus and the ulna and the radius, for instance, but then they have lots and lots and lots of finger bones packed together. So ichthyosaurs superficially resemble dolphins, and that's because they developed very similar behaviors. They swim through the water very, very fast. Presumably they would, jump out of the water like dolphins. Again, they've been extinct for a long time, but they probably behaved very similar to dolphins. And because of that, both dolphins and ichthyosaurs developed the same body shape because that's the shape that fit their lifestyle. This is called convergence because we have two very different organisms that converged onto the same strategy. They converged onto the same structures, or in this case, the same body shape. Let's have a look at some more examples of convergence. And again, that's where we have two fairly distantly related organisms. They don't share a recent common ancestor that start to resemble each other over time. And it's just due to independent evolution that causes them to adapt a similar lifestyle and develop similar appearances and structures. We're going to look at mammals this time. There are two big categories of mammals. We have the placental mammals, and they're like us. They carry their babies to a more developed state than we see in the other group, which is the marsupials. Marsupials give birth when the baby is still quite underdeveloped compared to placental mammals, and then the baby crawls into a pouch and feeds on milk. Let's go back to our continental drift model here. This is where this stuff is going to get important. So remember, back in the Permian, we had one massive supercontinent, that was Pangaea, and then that started to break up in the Triassic. You can see at this point, it's broken up into two large pieces, Laurasia and Gondwanaland. And things continue to break up through the Jurassic and the Cretaceous. And of course, by present day, we have the continents very widely separated for the most part. When this breakup was occurring during the Triassic and the Jurassic, that is when placental mammals and marsupials diverged from each other. They separated out into two major groups. And what happened, and I'll try to draw this on the Cretaceous map, is the marsupials found themselves isolated in South America, Antarctica, and Australia. And you can see those three continents are all connected together. Those northern continents had marsupials and also placental mammals, but the placental mammals outcompeted the marsupials and pretty much wiped them out. But they were safe on these three southern continents that stayed stuck together for a while. Australia went its own way, separated off completely, and the marsupial animals that were found in Australia were safe from the placentals. The placentals had no way of reaching Australia, well, until humans unfortunately helped out with that. But anyway, as you probably know, most marsupials are found in Australia, and that's the reason. You might be interested to know that there are fossils of marsupials in Antarctica. It's a very cold, icy place now, but it was tropical at one time. There's lots of fossils there. They're just really hard to get to because they're under lots and lots of ice. South America had marsupials as well. They did fairly well, but they got wiped out around the time of the last ice age, except for one example. The possum, or opossum I should say, went north and it moved up into North America as well. The problem with South America is when it joined to North America, the placentals came down and they wiped out everything except for the opossum. But anyway, the key point here, in Australia, we had marsupials that were isolated from placentals. They didn't have to compete with them. They did just fine. And they evolved into lots and lots of different species. And they took on different lifestyles. The placental mammals that were located on the other side of the world in some cases took on similar lifestyles. And they developed very similar structures and appearances, even though they have been separated for a very long time and don't have a very recent common ancestor. So we have moles that are very well adapted for digging. They've lost their eyes. They have massive big front feet for digging. And we see the exact same adaptations in placental moles and marsupial moles. We have anteaters that look quite similar to marsupial anteaters. We have mice, we have placental mice that look quite similar. Again, all the placental animals have a pouch. That's what defines them. We have lemurs and we have spotted cuscus that look very similar. We have flying squirrels and we have something very similar in marsupial form as well. We have bobcats and we have a marsupial cat. They look very similar because they're hunting similar prey. The flying squirrel and the marsupial equivalent, they both evolved that habit of gliding from tree to tree. Again, they did that totally separately. And then we had a wolf, and we had a Tasmanian wolf, which unfortunately is extinct now, very much like a placental wolf, but it had a pouch. So again, when we're trying to figure out relationships, we have to be very wary of convergence. We have to try to identify it and ignore it as one of our characters. And here's that marsupial wolf, the Tasmanian wolf. Unfortunately, they were hunted to extinction. This is the last photograph of the last known Tasmanian wolf that died in a zoo in 1936. Occasionally, there's people that claim they've spotted one in Tasmania, an island off the southern coast of Australia, but unfortunately, very unlikely. You can see how similar the Tasmanian wolf is to the North American wolf, especially if you look at the skulls and look at the teeth. And that's because they're catching similar prey and they have a similar lifestyle. But to find a common ancestor for these two, we'd have to go back at least 150 million years. Here are some moles. We have the golden mole, which is a marsupial. It has a pouch. At the top there, we have the North American mole down beneath that. Notice that they have very, very short velvety fur, so it doesn't trap too much soil as they're burrowing through the ground. They have very large front paws to dig. They're great big shovels. They have extremely reduced eyes to the point where you can't see them through the fur. But again, this is an example of convergence. They just happen to evolve the same lifestyle. And this is the form. This is what an underground mammal should look like, because this is what works best. But they arrived at that independently. So we always have to ask ourselves, if we come across shared derived characters, are they the result of shared ancestry? Are they homologous? That's what we want. That's what we're looking for. Or are they the result of analogy? Taking a look at this figure on the left, and I'm going to just focus on the first one there that shows the structure of a wing in 3 different organisms. We can see that the shape of the wing is very similar. And in fact, it's kind of similar to the guy in the wingsuit. And that's analogous. The wing has to have that shape in order for flight to occur. But now we look at the pattern of the bones. The pattern of the bones is due to homology. We have to go back quite a ways. We have to go back to the appearance of the front limb in our distant ancestors, but that is due to analogy. You can see that these 3 different animals solve the problem of flight in very different ways. The pterodactyl is kind of my favorite. It's got one very, very long digit that supports a membrane. So that one long digit supports this membrane that's used for flight. In the bat, we see something kind of similar where all the fingers are stretched out and they support the membrane. And then in the bird, of course, we see something a little bit different. We've got the bones of the hand fused together to make some rather odd shaped bones, and we've got feathers, of course, to help out. But again, The shape of the wing is the same, that's analogous. The pattern of bones is the same, but that's homologous. Another thing we have to be very careful about if we want to make proper groups, proper clades, we need to take the animals that we've decided belong together and should be put into this new classification. We have to trace all of them back to one common ancestor, and then we have to make sure we haven't left anyone out. So in yellow there, we have a good example of a nice clade. It's a nice clade because it's monophyletic. So we have all of these different things here. We have turtles, we have crocodilia, we have the birds and so on included within this yellow group. And we can trace all of that back to the node that is labeled reptilia. That's a good clade if we define it that way. That's not the way it's usually defined. People don't usually include birds, aves, within the reptiles, but they should be if we're doing things right. People usually define reptiles as what you see in blue. They exclude the birds. That's not right. That is not a proper clade because we have left out a group. If we have most of the members in there, we have most of the members clustered the way we should, and we can trace them back to a common ancestor, but we've left out just one or two individuals, we refer to that as a paraphyletic group, or a case of paraphyly. That's bad, we don't want to do that. Worse still is polyphyly. So a polyphyletic group is where we take two individuals that are very widely related. They're not closely related at all. And we try to put them together and we leave out all the other groups in between. Mammals, mammalia, and aves, birds, we might be tempted to put them together because they're both warm-blooded. That means they can regulate their own heat. That's a bad choice. There's lots of other things that obviously make them very different. If we put those two together, and that's what you're seeing in red here, that would be a polyphyletic group. Again, we're taking distantly related organisms, we're bringing them together, we're leaving out most of the stuff in between. You can see if we want to find a common ancestor for the mammalia and the aves, we have to go all the way back to amniota, and we should include all of that stuff that's in blue as well, but we're not. So that's a bad group. So some more examples here of what we don't want to do. Let's say we've decided to group A and B together. That's a bad idea, because if we take A and B and look for a common ancestor, we have to go all the way back to node 2. If we work up from node 2, we'll see that we should have included C, but we left C out for some reason. So that is not a proper clade. That is not a monophyletic clade. It's a paraphyletic clade. Likewise, with our second group here, we've included G and H together as a group. And if we decided to only go back to node 4, that'd be okay. But we've decided for some reason that we should go back to node 5. Well, we haven't included E and F, and we should, obviously. So with a paraphyletic group, you've grouped together some organisms that are closely related. You've almost made a clade, but you've left a few things out. With a polyphyletic group, you are taking organisms that are very distantly related and trying to bring them together. So here you can see A and B, they're fairly closely related, but we're trying to include E and H as well. And E is very distantly related to A and B. H is very distantly related to A and B. This doesn't work. And in fact, if we wanted to go all the way back to our common ancestor, we'd have to go all the way back to node 7. And if we did that and we wanted to make a monophyletic group, we of course would have to include C, D, F, and G as well. Another real-world example here. We've got turtles and we've got lizards and snakes, which are closely related. It's thought that snakes are perhaps lizards that have lost their legs. That's one theory. We have birds here right in the middle. We have non-avian dinosaurs, which is really neat. That's how dinosaurs are classified sometimes. It literally means non-bird dinosaurs. Or sometimes birds are classified as living dinosaurs. And then we have crocodiles. So we have this relationship here, as you can see on our cladogram. Now let's form some groups. We could do this, and this is what people would generally consider as reptile. If you tell someone, you've got reptiles at home, you've got turtles or lizards or snakes, maybe a crocodile, not a dinosaur, but that would be included in that group as well. Most people would not consider birds a reptile, but obviously they should be. We're going all the way back to the bottom of this cladogram to find a common ancestor. Working up, we should include birds. This is not a good clade. So the word reptile, we use it all the time, biologists use it all the time, but it's not a proper formal taxonomic group. We could divide things up like this. Now that would work. We've divided things up into smaller groups, of course. The Archosauria, we could do that. We have to include the birds, of course. So this works okay. Or we could do something like this, the dinosaurs. This is the dinosaur group. By the way, did I mention that Sir Richard Owen coined the term dinosaur? I'm not sure if I did. Anyway, dinosaurs include all the dinosaurs you love, Stegosaurus, T-Rex, etc., but should also include birds because, of course, we have to come back to that one point. We work our way back up so we can see all the descendants from that point, from that node. We have to include birds. Let's take a look at one more real-world example here. Towards the left, the first two groups there, those are fish. We'll talk about those later. Then we've got amphibia, we've got the mammalia, the mammals, and we've got our traditional reptiles towards the right there. Let's do something really dumb. Let's pick a really silly characteristic to form a clade. We're going to put together snakes and something else you might not have heard of, caecilians. Snakes, of course, have no legs. They're very long and skinny, and they have no legs. And caecilians are very similar, except that they are amphibians. They're slimy and moist. Snakes are dry and scaly, and very different from amphibians. So these are two groups of organisms that evolved the same shape and the same kind of way of moving because it worked. It was beneficial. Now, if we decided to put those two things together, we decided for some reason that leglessness was really important, and that's the characteristic we're really going to focus on looking at these two groups, and we tried to put them together, we would have a problem. We would have a polyphyletic group because we have to go all the way back to here, to this node, to get our common ancestor. And then look at all the stuff that we're leaving out. So we have two groups that are very distantly related, and we've left a lot of stuff out. That is kind of the defining feature of polyphyletic groups. Another interesting and also rather nerdy example here. What I've got in the red box is what most people would consider fish. We've got things like hagfish and lampreys and sharks and rays and sturgeons and bony fish like perch, etc., coelacanths, lungfish, all things that I think most people would agree, okay, that looks very fishy. So those are fish to most people. Now, the term fish is an informal term. Everyone knows what you're talking about when you say fish. But what if we decided fish were a clade? We would have problems. We'd have to go back to our common ancestor, which would be this node labeled Craniata. And I should mention the labels that are put there, those are the labels for the big groups that come out from that node. Anyway, we have to trace back to our common ancestor, which is that node that I've shown as a red dot. And of course, we would have to include all the stuff to the right. And you know what? We're in that stuff. We're in the mammals there. So technically speaking, for a taxonomist to be using the term fish, he would have to consider himself a fish as well. And I kid you not, this is a common nerdy joke. You'll see two taxonomy people pass each other in a hallway and they'll call each other fish. Another thing we'd like to avoid, if we can, is something called a polytomy. We know that we like dichotomies. That's where we have one node that splits into two branches. A polytomy is where we have one node that splits into three or more branches. So let's take a look at the cladogram here. It's for plants. We have green algae, not actually plants, that's our outgroup. We have bryophytes, those are mosses. We have tracheophytes, those are ferns. Gymnosperms, those are conifers, so evergreens. And then angiosperms, those are flowering plants. And you can see that we have a polytomy here. So we've run a whole bunch of data through the computer. It spat this out. And what it's telling us is it doesn't know how to resolve this. It's not sure if ferns are more closely related to gymnosperms, or if gymnosperms are more closely related to angiosperms. It's not sure where to put the gymnosperms there. This can be kind of exciting if you come up with this, because it means you've got some work to do. I mean, science would stop if there was nothing to learn. We've got something to learn here. We have to figure this out. But the end goal, when we're all done, is to try to resolve that node and turn it into dichotomies. I think for our purposes, this is probably the simplest cladogram format to use. So if you have a question on an assignment or a test where you have to actually draw a cladogram, use this one. Down towards the bottom left, we have the root of our cladogram, and that's where our common ancestor is for everything on the cladogram. We have species coming off, as you can see, as branches, and the red lines indicate where a new derived character pops up. Typically, when we go from left to right, we're adding more characters. And in many cases, that means that the organisms are getting more complex, but that's not always the case. We're adding more derived characters. Now remember, a derived character could also be a reversal. It could potentially be the loss of something. And our concepts. So taxonomy is the science of classification. Doesn't have to involve living things. Traditionally, it was based solely on similarities. So think of Carl Linnaeus, that's what he did. But now we base it on shared ancestry. And when we do that, we refer to that as systematics. Cladistics is probably the most common way to engage in systematics, where we're trying to figure out phylogeny. In a cladogram, new taxa, so new groups, new clades, we should call them, appear through dichotomies, never polytomies, if we can avoid it, and all taxa are represented as the terminal branches of the tree, so they're at the very ends of the branches. In cladistics, all taxa should be monophyletic. If they are, we can refer to that taxa as a clade, but it has to be monophyletic, which means we can take all the members of the group, trace them back to one common ancestor, and we haven't left out any of the descendants. And keep in mind, despite our best efforts, we never will truly know the phylogeny of any organism. We have to use logic, we have to use Occam's razor, which is looking for the simplest explanation to try and make sense of the data. And down the bottom here, I've got a few examples of clades that are good and taxa that are not clades. And finally, our terminology, important terms that you should know. Stay tuned. Next time, we're going to talk about viruses, which are very fascinating and are not technically alive, and we'll talk about why that is.